Good morning. I'll be talking today about using molecular epidemiology to investigate environmental exposures in pediatric cancer. So many of you are familiar with the molecular epidemiology paradigm. What we're trying to do with molecular epidemiology is open up the black box between exposure and disease. Some of the exposures that we're interested in for childhood cancer include infections, parental tobacco use, ionizing radiation, pesticides, and diet. Now, what do we look at in diseases to try to connect with these exposures? You've heard today already about mutational signatures. Today, I'll be talking especially about gene deletions in childhood leukemia and also DNA methylation changes. Another topic of today's lecture will be using DNA methylation as a way of measuring exposures, or at least effective doses of exposures. So in the past 70 years, really, leukemia rates have been rising. Here's data from a couple of publications up to 1992, and the first one on the left, and in more recent years on the right. And what these publications have seen is that there's been a 35% rise in leukemia rates, childhood leukemia rates since 1975, effectively a 70% rise since World War II. What this means is that environmental causes have to be behind these rises, since we haven't really changed our genetic code over these years. Now, our work in the past years has really focused on looking at environmental exposures and etiologies at two particular time windows in childhood development. That is during pregnancy, where we know that there are initiating lesions for childhood leukemia that are present at the time of birth, and early in the child's life, where secondary genetic changes occur, which then cause the eruption of the leukemia as a disease. And there are certainly genetic modifiers involved here. I'll focus specifically today on environmental modifiers, the infection and tobacco exposure. So childhood leukemia is a disease of the immune system, so it's not a surprise that infections should be involved. But it's long been a question, what infections? What we've done with childhood diagnostic leukemia samples is RNA-seq of the leukemia cells, and also DNA sequencing of plasma virions in children with leukemia to try and discover if there are any specific infections involved. We compared lymphocytic leukemias to myeloid leukemias and found that one virus in particular was present in the lymphocytic leukemias and almost completely absent in myeloid, and that's cytomegalovirus. Now, the question is, CMV is kind of ubiquitous infection. Most of us are carrying it. I know I am. So maybe it has something to do with the timing of infection. And in order to address that, we looked at neonatal blood spots collected at the time of birth and looked for cytomegalovirus sequences within those blood spots. And what we found is that children who grow up to get leukemia are much more frequently positive for CMV at the time of birth, indicating that there is a vertical transmission from the mother. And here is a control. We looked at Epstein-Barr virus, which is associated with other hematologic malignancies, but does not seem to be involved here for lymphocytic leukemias. Since that time, we've tried to assess how CMV is involved in leukemiogenesis. When you get it early here in life, it seems to change your immune response to other infections, leading to childhood leukemia. So we have a more vigorous response to other infections of children in children that are positive for CMV at birth. Another subtype of leukemia called the high hyperdiploid leukemia does not have this association with infections after birth. And in those leukemias, it appears that CMV may be directly involved in causing the leukemic phenotype or the disease itself. Now switching to tobacco and acute lymphocytic leukemia risk. Tobacco has long been of interest, especially pregnancy-related tobacco exposure, maternal smoking during pregnancy, although it's never been tightly linked to childhood leukemia. But we've asked the question whether tobacco may fit this developmental origin of disease hypothesis, where tobacco may cause epigenetic switches in specific genes. And one reason for looking at tobacco is a molecular genomic feature of leukemia cells, which are gene deletions, which are commonly found in lymphocytic leukemia tumors. Now, what I'm showing here is a study back from the 1990s where Barry Finette and colleagues looked at HPRT marker mutations in mothers who are smokers versus mothers who did not have tobacco exposure during pregnancy. And what he found was that the T cells from mothers that were smokers, the T cells of the children, and these are cord blood samples, the cord bloods often had these VDJ recombinase mediated deletions in tobacco exposed children, whereas there was a lower frequency of these deletions in non-smoking mothers. So this led to a hypothesis where tobacco exposure may lead to these off-target recombinase mediated deletions. And what I mean by off-target is in genes that are specifically associated with leukemia in families that have smoking parents. So the particular genes of interest are these. These are genes known to be deleted in lymphocytic leukemia patients. So we assessed these genes as deletions in 846 tumor samples from our California childhood leukemia study. And what we found in a case analysis, this is comparing cases of smokers to, smoking mothers, smoking parents, and to cases from non-smoking households. And these are risk ratios here, showing that when there is the presence of smoking, especially at the time of breastfeeding, here you can see where the largest associations are found. There is a higher frequency of gene deletions, which really supports this hypothesis that tobacco may be involved in causing VDJ recombinase mediated deletions. Now, in this study, we had active participation or questionnaire information about smoking from the families. We have another study in lymphocytic leukemia patients with 4,000 cases and 4,000 controls where we do not have questionnaire information. So here, what we did to assess smoking in newborns was to look at a DNA methylation mark for maternal pregnancy smoking. Here we have an EWAS study of maternal smoking showing very strong associations with particular DNA methylation marks in cord bloods of children of maternal smoking mothers. So we developed an inexpensive assay to assess smoking in 8,000 subjects in our study. We can't run these methylation arrays on 8,000 people in any kind of economical fashion. So we use this rapid, inexpensive droplet digital PCR assay. Here are some results. This is a gene by environment WAS type analysis where we assessed environment effects, so tobacco effects on genes. We also assessed gene effects on the disease. Here is lymphocytic leukemia, and G by E effects. And here we have the top 10 hits in this analysis. Here I cross out 4 of them because these are gene-only associations. So there was no environmental smoking effect or G by E effect. A summary again of these 10 hits. These 4 are gene-only. You can see that the disease gene p-values are very extreme for those gene-only results. We have a few other genes here, such as XOC3 and CDC162P, which seem to be environment only. So the environment disease effect is very strong for those. And here's the G by E effects over here. There's only one gene, PTPRK, which in this analysis came out to be a significant G by E effect. So just to summarize, we've looked at today how molecular measures can be used to assess risk and also genetic features within the tumor itself, those gene deletions, where both environmental exposures can affect DNA methylation, and also how we can use molecular measures to assess infection in the etiology of leukemia. And thank you very much. I'd like to thank all the group.